Okay. Uh, yeah, I'll start about the Javan tiger story again. So Java is an island in Indonesia, and it used to have tigers about 50 years ago. Okay, since 50 years, it is said that the Javanese tigers are extinct. However, recently, just a few years ago, this photo was taken in Java, and in this photo, it is very difficult to figure out, obviously, whether it is a tiger or a leopard. But just about a few years ago, again, there was a villager in the west of the island of Java who reported seeing a tiger, and the people from the Wildlife Services Forest Department interviewed this villager and showed him photos of leopards and tigers just to know if it is a leopard or a tiger the villager saw. And the villager was confident that he saw a tiger and not a leopard. So there was a huge survey done, and they collected one strand of hair from a fence along that part, and that strand of hair was brought to the lab. Okay, then they all then the researchers, they went to the museums, where the museums already have skins of dead Javan tigers. So these are tigers that were shot by the colonizers, the Britishers, and everyone else, and these were preserved in the museums. And they got some skin samples from the Javan tigers in the museum, and they have this one strand of hair from the forest. Okay, and what they're trying to see is if this one strand of hair is similar to the skin sample from Javan tigers from the museum, then they would know if it is a Javan tiger in the wild or not. And they did the analysis, and what they find is, oh wow, the hair sample matches the skin sample. So it must be a Javan tiger, right? And that's what they thought and published. However, when we redid the analysis, what we found is that they have analyzed the wrong sequences of DNA. So when comparing, uh, so when you compare two individuals, you want to compare the same DNA strand. Okay, you cannot look at chromosome one from individual one and chromosome two from someone else and say these are similar or different individuals. Right? You have to look at chromosome one for both. Similarly, what they did here is they analyzed something known as a nuclear copy of mitochondrial DNA. So they analyzed nuclear DNA and compare it to mitochondrial DNA. And then they thought they hit the jackpot, but that is not the case. So the analysis was a bit flawed. We redid the analysis, we redid the experiment actually. The entire experiment was redone. And what we find is the hair strand indeed belongs to a tiger on the island of Java, where tigers have been extinct for 50 years. So there is a tiger on Java, but we do not know what tiger it is. So there is another island next to Java, it's called Sumatra, where tigers live, and Sumatran tigers are extinct. And we do not know if the hair strand belongs to a Sumatran tiger or a Javan tiger. Okay. Now, if it was a Javan tiger, we would have known that Javan tigers are still there, and a proper conservation plan could have been formulated. We could have found out if it's a male tiger or a female tiger. We do not know that. We do not know if the tiger swam from Sumatra to Java, which is an unlikely scenario, but could have happened. There are also pet tigers that people have. There are tigers in circuses that could have escaped. So there are a lot of ways a tiger could have ended up in Java. But in either way, in either case, if it is a Javan tiger, it would have been such a great news, right? Because we thought this tiger was extinct. But we do not know that at all. So genomic analysis will help in this scenario. So if we can get the whole genomes, we can talk about whether it is a Javan tiger or a Sumatran tiger. So what does genomics tell us? I'm sorry, this is Indonesia. There is no genomics happening. So I presented you studies of all these fancy scales from the West, from UK, from America, from India, in NCBS. And there are all these nice studies. But when it really mattered in Indonesia, where genomics could have really helped, a genomic study could not be done. Why is this not possible? What is happening? Why can't we do genomics of these samples? Tigers are a global symbol, right? Everyone should be pouring in resources to actually do this. But in reality, what is happening is, if you look at this map, every dot represents one wildlife genomic studies, and you will see these dots mostly come from Europe, from North America, but there is almost no studies from Africa, South America, India, or the rest of Asia. Actually, there are some in Japan and China, of course. But mostly where the wildlife species are, no genomic studies are happening. So there is no capacity. So what are the challenges? First of all, the samples, they need to be non-invasive. What that means is we need to think, we cannot always get blood samples. You cannot go into the wild and think about getting blood sample from a tiger. It's illegal to touch a tiger, and there are many good reasons to not do that, right? So we have to think about innovative ways of getting samples for genetic studies. We have to get DNA from places that is not thought of. For example, it can come from a water sample, it can come from breaths. I'll get into that. Also, the resources that are needed, the sequencing facilities needed. You also need a lot of computers. Okay, I'm talking about big data here. All of this analysis happens on a supercomputer. Sometimes there is no supercomputer in the entire country for genomics. There is supercomputer for defense, there is supercomputer for medical research, sometimes for agriculture research, but not for wildlife conservation. So what are the non-invasive samples that have been used? So there is shed feather, there is broken eggshells, there is shed skin, for example, from snakes, there is fecal samples. This is a whale dandruff. Okay, whales are mammals, they shed dandruff, and you know whales are huge, so their dandruff can look huge. These are called skin sloths. And the deers, they shed their antlers, and these are non-invasive because you don't have to catch a deer to get DNA from their antler. So these are called non-invasive sample. For any of these samples, you don't have to actually interact with an animal or even see it. You can go, you can collect these samples, and you can get DNA from it. There are a lot of advantages to such kind of samples. They're accessible, first of all. So for example, if you look at this graph, you will see that shed hair happens to be the most abundant sample, then scat and carcasses, which are dead bodies. These are also non-invasive because you're not actually touching a live animal, are the rarest. And the rarest are blood samples from alive individuals, which happens after tranquilization. They're very rare to come across. Okay, there is minimal interaction, there are huge number of samples, and there are very few ethical issues, right? Like, it's okay to take a portion of the tiger scat out of the forest, but it's definitely not okay to touch a tiger, obviously. So you can sample and you can potentially get DNA, and you can do this continuously over time with minimal disturbance to the wildlife. Of course, there are some disadvantages like contamination.